Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, the final speaker for this session is Jinyi Cha from Queensland University of Technology. She'll be talking about making sense of sequences, comparative genomics, and the new GIS.
Hey, so thank you very much for joining me. So my name is Singy, and I'm from the Microsoft QUTE Research Centre from Brisbane, Australia. So we're in the bioinformatics department. We're developing tools and applications that will help scientists in making sense of sequences, and in the area of comparative genomics and the new GIS. So a quick overview of the talk today. I'll begin with an introduction of the difficulties that current biologists are having, and introduce the Bio 2.0, so our solutions in how we're going to attempt to overcome these difficulties and address the problems. So bringing in the GIS to comparative genomics, and conclude with our plans for the future. So with the new and improving development of sequencing machines such as 454, we've got an explosion in the genomic data that's available now, especially in the last 10 years, illustrated in the graph there. So what we have there is looking at sequences and scale of the number in millions available that's in the GenBank database. So basically it's the rise of population, genomics, and proteomics. So it's great that we're having all this data available, but what do these sequences mean? When I find a new gene, what's the function of this gene? Why do I have one species that's so alike to another, but what is it that's making them so different? So these are the type of questions that biologists want to answer when they're trying to make sense of these sequences. And a common approach that they use is through comparative genomics, where they take a region of the DNA or protein sequence and make comparisons against each other across a number of genomes. So I just want to use a simple example, so we're starting from common ground. This is a common approach used when looking for similar genes, trying to do protein characterization, so trying to find out the function of a particular gene. So I start off with my gene of interest there, put it through BLAST, which is the common tool that's available from NCBI, so that's a screenshot. And what BLAST returns is a list of hits, with each one with an associated e-value. So that's a list of similar genes. So this is an example output. What I'll do is okay, so that's clear up. So at the top, you have a summary of the results that you get. And here is a list of the results. Each of them have a name of the gene, and as I mentioned before, the associated e-value score there. If we keep scrolling down, for each one of these hits that we get, we also have more information and the actual alignment view, showing the actual position, similarity, and differences back with our starting gene. So that's, again, summarized in this picture. This is the result of one gene comparison. Now, let's look at something that's in the tens region. So we start to get a lot more alignment. And imagine if we're doing an m by m-1 gene comparison against a number of organisms. So a specific example is for my own PhD research, where I'm going to be studying Yersinia pestis, which is the cause of plague. Not a very nice little bug there, against E. coli. And both of them have over 4,000 genes. So you start to get the idea that it's information overload, and it's just too hard to comprehend in the text-based output there. So I want to make a note that we do understand that this text-based alignment is important to the biologists, but prior to stepping straight to this alignment, let's take a step back and let the scientists visualize it in a graph. We want to have a look at the distribution of the genes that are similar to what we started with. So we make the topology fit the question. And we take advantage that visualization is the key to navigation. So what I mean by that is there are certain details or patterns and clusters that you can see that you can spot easily when you're looking at a graph, as opposed to the previous example where it's all text-based. So this can lead the scientists to refining and testing their hypotheses. And when they reach an answer that they're happy with, or they come to a new discovery, we provide them with functionality for them to share their views with their peers or with a collaborator that's situated across the globe. And we provide this with more than a static image and more than the print screen button. So I'll demo this in a few slides. So the beauty of our approach is that we're using a concept that's been around for a long time. It was developed before even I was born. So the idea of that is that the users don't have to learn something completely new. So we're familiar with the GIS. We're comfortable. When we look at maps, we know the cities as dots on the maps, and when we think of maps, we think of distances. But when we apply GIS to our application with comparative genomics, the dots there are genes, and the distances represent the similarities between the two genes. So in addition to that, we also linked to a collection of other data sources through Bio2IDF. And this is our new GIS. So this time it's the Genomic information system. So I've been talking about our system and what we're trying to address here, and I haven't even mentioned the name. So our system there is called SilverMap, so named because it's been implemented in Silverlight. Silverlight is client-based, so it's fast. All we do is send the data across, and all the rendering happens on the client side. Silverlight also provides a richer environment, so we make use of this in implementing animations, which will help in data comprehension for the scientists, and this will become apparent in the demo. So here's a schematic of our model. SilverMap is linked to the Bio2IDF repository, which is also used with tags, so users can put their own annotations in there. Our first one is the BLAST component, which we have a local copy of, but essentially we will have it link up to NCBI BLAST, as well as sometimes scientists would like to perform their own BLAST on their local machine, so we'll have that implemented and supported. Now, I keep talking about distances for users and using the BLAST as an example, but essentially our goal is so that the user can provide their own defined distances and implement that, and that will take over the BLAST component. And so far I keep talking about how distances represent the similarity between genes. Another example where the distance between two genes actually doesn't mean anything is in transcription regulatory networks and modeling protein-protein interactions. So what we have there is the distance between two genes don't necessarily mean anything, but the directed edge from one gene to another is significant because we want to capture that relationship in there. So another advantage of Silverlight, as I mentioned previously, the animations and the ability to dynamically highlight the sub-networks of these two models, again, helps data comprehensions for scientists. So without further ado, I'll have a look at the demo. So just coming back to this example BLAST output here, which I've already done previously, it's showing the output of one gene and finding similarities of that gene. But what we don't get from this output is that how closely related are these genes to each other. So I've done that using our local BLAST database. So these are the results, and I did the BLAST. And just keeping a traditional approach, these are the actual sequence-based alignments. But we'll jump over to SilverMap. So this is SilverMap. We're keeping the sort of familiar interface that you'll see with maps and navigation buttons. And what we have in the center is the gene that I'm interested in. So I can zoom in just using my mouse, scrolling in. So this gene is found in Yersinia pestis. It's sort of known to play a role in virulence, but we don't know its particular function. So the idea is to find similar genes and try to infer a function for this one that I'm interested in. Now, I've mentioned about the animations. So if I don't like this reference gene, what the distances that I keep mentioning about is in relation to all these ones on the outside. So distance, the radial distance between the nodes on the outside to the center there. So that's been represented by the E value, which is down here. So what I can do is, if I don't like that reference gene, I can change the view by double-clicking, and that navigates to the center. And everything else models around it, so I can get that immediate response and I can analyze the data. So another animation there is that each individual sort of thinks differently depending on the type of animation. So in this one where we select the reference gene that we were interested in, it jumps to the center and everything else navigates around it. I myself don't particularly follow that quite well. So what we can go on the fly is where you select your gene and everything else moves to that. So you've got all these different types of animations there for the user, and that again helps them comprehend their data. So I'll come back to this one. So again, I was saying in the beginning that I'm trying to infer the function of this gene. Come over here. But what I want to do is to just focus on the genes that are really close to this one. So what I have is this radius, which is a threshold cutoff, which will dynamically move that red dash circle. So I just want to focus on the genes inside that red circle. And down here, I've got a summary of all the genes that are in that circle, which tells me quick notes about them. And if I just zoom in, so each note there has a label, and those with the asterisks have an associated tag that's been previously annotated. And let's just have a look at what this one has. So the tags, it's a membrane protein. So I'm just going to copy that with Control-C. I'm going to go in my tags. So in SilverMap, we also have tagging where we can use to select subnets. So these have, the ones that are highlighted in blue have all been tagged with E. coli. So the one that I was interested in is membrane protein. So we already know that these three are actual membrane proteins. So what I can do is make an inference that maybe the ones all there in the center there could be membrane proteins. I just want to tag all the genes in the center. Just apply the tag. Okay, they've all been tagged, so the asterisk is coming up. There's my, and it appears down here with a count. So previously one last thing is that I was mentioning how we can collaborate and share this view. So if the scientist is happy with this view, what they can do is the ability to come down to the HTML screenshot panel, which produced this div object. I can select all that, copy it, copy that, and then go to my virtual lab wiki page or a HTML, put that into the HTML, and what it will produce is a widget mode. So this is an actual live HTML page. So it's about Yersinia pestis, the bug that I'm working on for my PhD. But what we have down here is the actual widget mode of SilverMaps, and it's fully operational. So you get the zooming, different navigations, and you can also have set different thresholds. So you can simply do that, configure your web page, use the URL, and then send that off in an email to your collaborators. They come to this page. It always loads on that default. Okay. So just to conclude, future directions, where we're headed. So as I mentioned before, it's not just about similarity between genes. Ideally, we want the user to come up with their own distances, and we want to support that. We want to also look at modeling transcription regulatory networks and protein-protein interactions with SilverMaps. And basically, so bring our tools to the community. So being able to make SilverMap the lingua franca of genomic publications, as I mentioned, demoed with the virtual lab book and the public website. So integration with tagging and sharing of views. And just one quick demo that what we also tried in the previous week was because scientists are so used to Excel and spreadsheets. So we looked at, a member of our group looked at the possibility of integrating the Silverlight component into Excel. This is another one of our tools called SilverGene. It's also implemented in Silverlight. It's more mature than our Maps tool. But basically what this is showing is you've got, you can load up different genomes. So it's a traditional genome type browser. I can narrow down to sequence views, but it's linked. So I can change the view of the window on my Excel form, and it will dynamically propagate that to the genome there. And I can add different tracks to this. And again, it will come up. And the user has all the configurability to change the view and move about. So same idea there will apply to SilverMap. So we look into the ability of incorporating that as well. Here we go. Hey, so last slide, just like to thank Queensland University of Technology and funding from Microsoft Research and Queensland State Government, and Microsoft Research giving me the opportunity to come and represent my group and give an introduction of what we've been working on. And thank you very much for listening.
Thank you very much. We have time for a couple of questions.
So we've thought about using Silverlight, and I was just curious how easy, difficult it was? For us, it was actually fairly easy because it's a familiar C# environment. So we didn't have to learn a whole new language around it. It was a familiar environment to use. So yeah, it wasn't too difficult to sort of move over to Silverlight. Okay. It's Silverlight 1 that you used?
We started from the beta release. So we went from Silverlight 1, and then when the new one came out, we had to transfer everything over. So the one that I demoed there is the current Silverlight 2.
Okay.
Yeah.
Any more questions? Okay, I'll ask the usual one. Where is this available from? Can we download it? Can we use it?
That one is, you don't need to download. It's actually just on browser. So this is normal IE browser. Well, you just need to download the Silverlight plugin. But this one is a beta release. It's not publicly available yet, but we're releasing it soon in January. So you can navigate to it from our website.
So that's the QUT one that we can see in the toolbar at the top there?
Yeah, QUT, Macuta, QUT, EDU, AU. So it'll be out very, very soon.
And I think SilverGene's already...
Yeah, SilverGene's already out.
Okay. Okay, well, thank you very much. And please join me in thanking all the speakers.